I'm going to talk about some preclinical data that we have for AD214, and what we'll talk about in the next 10 minutes or so is some preclinical efficacy data that we've got on a bleomycin mouse model of lung fibrosis. We'll have some pharmacokinetic and pharmacodynamic data that might support weekly, perhaps even fortnightly dosing. We also show that in a GLP tox study that AD214 has a very good safety profile. I'll touch on the ability to label AD214 to use it as a PET imaging reagent, again to try and improve the information that we can get from the phase 1 trial. And I'll just talk a little bit as well about preclinical proof of concept data that were pending for other indications. So AD214, the precursor was AD114, and we had a lot of pharmacology data on that. So we then moved on to 214, and we had the FC portion of that molecule, and we built up then a very strong in vitro pharmacology profile of AD214, showing that it's got very high binding to its target. It affects the migration of PBMCs, which have CXCR4 on them, and it's able to modulate inflammatory mediators. We've also looked at an in vivo efficacy model. This is an IPF model, mouse model, and I'll talk a little bit about some in vivo toxicology data that we've got that shows that AD214 is very safe. So the bleomycin mouse model, as Tim mentioned, is a gold standard for IPF drugs that are trying to get into the clinic. Essentially what you do is you dose a mouse with bleomycin. That causes a damage to the lungs, and eventually that will cause deposition of collagen and lead to fibrosis. And so after 21 days you can see fibrosis in the lungs of these mice. What we've done is use the therapeutic model, so we give the mouse bleomycin, we allow fibrosis to occur, and then at day 8 we start giving our drug AD214. These are just some representative pictures from this model. You can see that the normal lung tissue, very nicely spaced airways with good blood flow. After 21 days of treatment with bleomycin, you're getting heavy blue staining here, which is indicative of collagen deposition and stiffening up of the lungs. And you can see that in the picture at the very end, that this is from a mouse that's been treated with bleomycin for 21 days, but at day 8 started treatment with AD214. And you can see that you really reduce considerably that deposition of collagen. And this is very indicative of being able to reduce the severity of fibrosis. This is some of the quantitative data, and I don't want to get into it in too much detail, but you can see that in the mouse model itself, naive mice have very little collagen deposition. 21 days of bleomycin treatment, you get very high collagen deposition, and we measure it by an Ashcroft score, it's called. Importantly, when we add to the mice a control antibody, an antibody that doesn't recognize CXCR4, that really has no significant effect on the deposition of collagen. We looked at pirfenidone and nintedanib in this assay, and again they do work, but they're not terribly good in this particular assay. And these two drugs are actually on the market for fibrosis at the moment. But I think importantly, at 10 mg per kilogram and 30 mg per kilogram every 2 days, or 10 mg per kilogram and 30 mg per kilogram every 4 days delivered to the mice, you get a significant decrease in the Ashcroft score, a significant decrease in the amount of collagen that's deposited in the lungs. So we think that this demonstrates AD214 has good efficacy in this model, which again is the gold standard, and it's supportive of perhaps a therapeutic window being as low as perhaps 1 mg per kilogram. We also did a GLP tox study, and we got some very important information from this. The elimination half-life of the antibody AD214 in the plasma of mice is about 22 to 29 hours. That's about 3 days or so in a human. Now this is just the AD214 in the plasma. What is that AD214 doing in the blood? If you look across here, this is receptor occupancy data. So we're looking now at whether AD214 is actually present on CXCR4, its target, in white blood cells. And I think you can see that 72 hours after we've looked at the mice, you can still see about greater than 60% receptor occupancy on the target. So we think this is supportive, for example, of a dosing window of about 10 mg per kilogram intravenously, weekly, and perhaps even every second week, because we're clearly getting extended pharmacology out to that point. The other important piece of information you get from a GLP tox study is the safety profile. And we've shown, and we've done 3 non-human primate studies. Again, we did 2 before this GLP tox. And in this one, we looked at 10, 30, and 100 mg per kilogram in multiple doses. And so 100 mg per kilogram is a very high dose. We found that AD214 was well tolerated. There was no deaths. There was no AD214 related adverse events. And there was no changes in a panel of clinical observations, which includes some of these that I've got on the table here. When we did see some changes, they tended to be very low, transient, and completely reversible within a few hours. And so we think that these tox studies were in line with expectations in the 2 previous non-human primate studies that we did. And we've done separate tissue cross-reactivity and cytokine release assays. And again, we see little toxicological significance of AD214 in those assays as well. So that's good complementation with the GLP tox. So we think this is AD214. This data means AD214 is really ready for a phase 1. But there is a situation at the moment is there's really not very good biomarkers to look at CXCR4, the target for AD214. And it's very difficult to confirm AD214 binding to the target in diseased tissue. So what we've looked at is to say, well, can we use this opportunity to try and take our AD214 and attach a radio label so that we can use it in PET imaging. As Tim said, we recently got funded to do this by the Medical Research Future Fund, and this project is on the way, and we've got a really good team of clinicians, chemists, and radio chemists as well. But I think some of the benefits that come from having a PET labeled tracer would be that we'd be able to confirm AD214 binding to CXCR4 in the diseased tissue in patients. We'd be able to get some idea of the duration and binding and tissue distribution of AD214 in patients. We may even get to visualize the severity of the disease in patients before they enter a trial. And we could also determine the impact of the disease on AD214 tissue distribution and availability. So we think this together will add significantly more value to any phase 1 trial that we do. So that's the preclinical data. What I want to talk about now just briefly is some other indications that AD214 might be useful in. And so the target for AD214 is CXCR4, the GPCR that Tim mentioned. It's important in maintaining stem cells in the bone marrow. It's also used by HIV as a co-receptor to invade T cells. And it's been associated with cancer. In fact, it's upregulated in around about over 20 different types of cancer. And of course, now it's recognized as a biomarker in fibrosis, not just in the lung, but in other tissues. And this is just some data that we got with our colleague Carol Pollock in Sydney, showing this is normal kidney in a mouse, showing you that there's no brown staining when we add our AD214, and so there's no CXCR4 there. But when there's a disease, when you induce disease in the kidney, you can see massive upregulation of the target. So this is a very nice situation to have where your target for your antibody is not present in the normal situation, but is present in the disease situation. We have this also in the lung, now we have it in the kidney. And so I think there's only one other drug that actually targets CXCR4 that's been approved, and that is Mozobil. It's sometimes called Plerixafor or AMD 3100. And that's only indicated for stem cell mobilization from the bone marrow. It's important to know that this drug was used for a lot of preclinical validation that CXCR4 was important in fibrosis, but it can't be used in the clinic because it's got a very strong toxicology profile. It's got a lot of adverse reactions, and so it can't be used in humans for chronic disease. And there's also several anti-CXCR4 biologics, particularly monoclonal antibodies, in development for cancer, but none at the moment in fibrosis apart from AD214, which we believe is the first in class. So we talked about the lung, AD114. We did a lot of preclinical evaluation of 114, and we've got a lot of that for AD214. But there's some things that we still have to do. We're trying to get for the lung fibrosis to look at AD214 blocking the migration of fibroblasts from IPF patients. We showed that that was the case for 114 with a colleague, Corey Hogeboom and Cedars-Sinai in Los Angeles, and we've got those experiments planned to do for AD214. Again, we don't think this is necessary for phase 1, but it's definitely data that we would like to have. We also have looked at AD114 and showed that it can block progression of fibrosis in the eye when you have various eye disease like macular degeneration, which is a very important side effect of AMD. And also, as I mentioned, we've shown that AD114 works in a mouse model of kidney disease, a folic acid model, and we're at the moment doing a mouse model of disease, a UUO, it's an obstruction of the ureter model, and that's being done right at the moment. The other areas that we're looking at is we have a small project on cancer to look at CXCR4 and AD214 in cancer, and of course I mentioned the diagnostic PET imaging reagent. So I think what we have is that we will have a phase 1 project with hopefully coupled with the PET imaging reagent, which will give us a lot of information that can then be able to inform the next steps. So do we go into a phase 2 IPF? Does AD214 go into a phase 2 trial in IPF or interstitial lung disease? Or is it could also be useful in phase 1b, 2a in, for example, macular degeneration, as I mentioned. It's also possible that it can be a diagnostic screening agent, standalone, and a phase 1b, 2a in the kidney, or phase 1b in cancer. And I think we either have preclinical data to support all of these, or some of that data is we're about to get. So I think the trick now is to tailor the ongoing preclinical program to try and best inform which of these areas that we could put AD214 into. And I think that's me. I'll stop here if there's any questions.